Motility of Listeria monocytogenes. Listeria monocytogenes is a bacterium that exhibits characteristic tumbling motility. This motility is facilitated by Poritrachus flagella, which are numerous hair-like structures that protrude from the bacterial cell. However, the expression of these flagella is temperature dependent. At room temperature, 20 to 25 degrees Celsius, Listeria monocytogenes is actively motile. In contrast, at body temperature, 37 degrees Celsius, the organism does not synthesize flagella and therefore shows less or no motility. To test for motility, a semi-solid motility agar containing 0.2 to 0.4% agar is used. The agar is stabbed with a needle containing the bacteria. At 20 to 28 degrees Celsius, Listeria monocytogenes inoculated into the agar column will swarm through the medium, producing a cloudiness that is easily visible to the naked eye. A layer of increased growth is typically observed about 0.5 centimeters below the agar surface, where oxygen tension is reduced, indicating that Listeria monocytogenes thrives in microaerophilic conditions. History of Listeria monocytogenes. Listeria monocytogenes was first described in 1924 by E.G.D. Murray in rabbits. It was initially named bacterium monocytogenes before being renamed Listeria in 1940 by Harvey Perry. While early reports of Listeria monocytogenes infection in both animals and humans exist, its significance as a cause of neonatal infection, sepsis, and meningitis wasn't recognized until 1952. Later, it was also associated with infections in adults with weakened immune systems, such as those undergoing immunosuppressive therapies. The link between Listeria monocytogenes and foodborne illness was not established until 1981, following an outbreak in Nova Scotia, Canada. This outbreak, primarily affecting pregnant women and newborns, was traced back to coleslaw contaminated with Listeria monocytogenes from sheep manure. This incident highlighted the importance of Listeria monocytogenes as a significant foodborne pathogen, leading to increased awareness and safety measures within the food industry. Poem on Listeria monocytogenes motility. A restless soul, Listeria, with flagella that twirl and stir, at room temperature's gentle grace, it dances with an eager pace. But warmth, like fever, stills its drive. At body heat, it cannot thrive. No whirling dance, no watery chase, its flagella hide, in dormant filth, in agar's depths, a cloudy trail, a ghostly form, a misty veil, where oxygen grows thin and slight, it blooms and thrives, in fading light. From Murray's lab, a tale unfolds, of rabbits young, and deaths untold, bacterium, then Listeria named, its history in science famed, a foe unseen, in shadows deep, in newborns' cries, and mothers weep, a danger lurking, in food we share, Listeria's threat, a constant snare. Yet knowledge grows, with every year, we learn to fight, and quell the fear. This tiny foe, with it in might, will yield at last, to science's light.